Now this is just the rates of reaction of a catalytic IgM that we isolated by screening patients with Waldenstrom's macroglobulinemia. These are patients who produce large amounts of monoclonal IgM, and we screened about 20 monoclonal IgMs and found one that hydrolyzes A beta as seen by HPLC and by mass spectroscopy assays. And this is a comparison of the Km. So this is an approximation of the binding affinity. It's the analog of Kd for a binding reaction. And Kcat is the rate of catalysis, how many molecules turn over per unit mass of the catalyst. So here are comparisons that the catalyst cleaves 95% of A beta using these assumptions shown here under certain conditions, and an antibody which does not have the catalytic function with the same parameters, with the same Km, would only bind 4% of the A beta. So there's little doubt that that antibodies such as this rather poorly catalytic IgM, so it's not a very impressive rate of catalysis, but nonetheless it is sufficient to anticipate a substantial increase in efficacy. Here we allowed A beta to form fibrils and oligomers, and then this was done in the presence of a catalytic IgM or a non-catalytic control IgM. And as you can see, this is done by atomic force microscopy. These fibrils are self-evident. These little dots are right here. They are the oligomers that are supposed to be the toxic species. As you can see here, in the presence of the catalytic IgM, there was a substantial reduction both in the amount of oligomers and the fibrillar structures compared to the non-catalytic IgM. This is simply an HPLC on the left side, a mass spectroscopic determination on the right side, by which we identify that the bonds that are cleaved in A beta are a lysine-leucine and a lysine-glycine bond towards the middle of the peptide. Now the ELAN antibodies that I mentioned are really directed to this region. I had mentioned that all antibodies discovered, naturally occurring antibodies with catalytic activity discovered thus far are serine protease type of antibodies. And by crystallography, our collaborator has shown that this IgM has a serine, an oxygen here, which is activated by hydrogen bonding with the side chain of an arginine, and then glutamate is the third component of a so-called catalytic triad, which is the hallmark of conventional serine proteases as shown for trypsin in this slide. And there is additional evidence that it's of the serine protease kind. We've shown this by both mutagenesis, site-directed mutagenesis, as well as by using specific inhibitors of serine proteases. The catalytic step is dependent on a serine protease type of activity. Now we looked at IgM and IgG from patients with Alzheimer's disease and non-demented age-matched control. And as you can see here, the catalytic activity of the IgMs is substantially increased in Alzheimer's patients. IgG is also increased. IgG, however, in general is less catalytic than IgM. Nonetheless, even here, the AD patients tend to have greater levels of catalytic activity. Here we measured the catalytic IgMs in plasma and in the cerebrospinal fluid. We find that the catalytic activity normalized per mole, or per microgram rather here, is greater in the CSF. So there's some evidence for local production of these antibodies within the brains. The take-home message is IgMs are better than IgGs. This is contrary to what is normally found, where IgG is the more adaptively matured, high affinity binding function of antibodies. It turns out IgMs are better catalysts. And then this increase here, it's sort of counterintuitive, but that Alzheimer's patients have more of protective antibodies. But I interpret these data to mean that the patients are trying to make more antibodies in order to protect against the amyloid. But it's probably too little too late, and these antibodies are not successful in controlling the amount of amyloid. Now this has become a pretty big story because, as I said, IVIG is now being used for therapy, and so whether patients with Alzheimer's disease have increased levels or lower levels for conventional antibody binding, it turns out that some groups say that the antibodies are increased, others say they're decreased, but the bottom line is that there is an attempt to link the body's immune defenses against warding off this dreadful disease. So these are B cells here from human B cells stained with CD19, which is the so-called B cell receptor, an antibody expressed on the surface in association with signal transduction proteins. And here, the green, we have stained with an electrophilic derivative of amyloid peptide containing phosphonate groups on these positions here. These are lysine residues. These are artificial molecules. They bind to antibodies covalently, and there's extensive evidence for that. They're also used as inhibitors of catalysis. But the more interesting thing in terms of autoimmunity is that they really are, they are sort of mimics of naturally occurring highly reactive dicarbonyl compounds that are produced during these aging protein glycation reactions. So bottom line, we synthesize these molecules as probes for catalytic antibodies because they bind covalently to the antibodies. However, these antibodies are naturally occurring, and there must be a stimulus for the synthesis of the antibodies as a function of age. So we have found that this is the antibodies increase with age, and we believe that there are naturally occurring analogs to these covalently reactive compounds that we create. Okay, so now the proposal that this can be used for therapy, and IgM, what is the basis of the proposal is what is depicted in this slide. And IgM cannot cross the blood-brain barrier efficiently. It's 900,000 Daltons, one of the largest proteins known to man and woman. It doesn't cross easily. IgGs get across far more easily, and IgGs, as I had mentioned earlier, the entry into the brain can be toxic. So we propose that pooled IgM from healthy donors, so the antibodies are found in undemented individuals as shown here. Alzheimer's patients just have a little bit more. We propose that this should be considered as a catalytic IVIG formulation to be administered intravenously, and this would work like a drain, a peripheral drain to remove A beta. The precedent for this proposal is the so-called peripheral sink hypothesis of Ron DeMatteis and Steve Paul, where they say that removal of A beta by binding in the periphery causes more leakage, more efflux of A beta through these transporter molecules at the blood-brain barrier. If you remove A beta from the periphery, A beta comes out from the brain. So IgM is well suited for that, and it's rather efficacious because it removes A beta more quickly.